So my name is Philippe Mercier from Swiss-Prot Group in Geneva, Swiss Institute of Bioinformatics. So I am a virologist by education, but since about 20 years, I've moved to bioinformatics and various ways of studying proteins and genomes, either by annotating in Swiss-Prot or by creating ViralZone, a website dedicated to virus structure and virus genomes from all the diversity of it. Okay, so today I'm going to speak about the new advance we have in structural modeling, that kind of a revolution in virology. Okay, so I wanted to share that. It's most of the resource on tools I will speak about are in 1 year old, and they are completely in development. So the course will be recorded, but maybe in 1 year it will be completely out of date because things are changing very fast. But there are very new things, and you will see that now the protein folding I kind of break the code, and we can make a lot of study and analysis based on structural features of viral proteins, which is huge for virology. Okay, so we have the course will be in 2 parts with a break between. I will make an introduction of structural virology, so why structure is important in virology, maybe more than in the cellular world. The modeling resource I'm using, so there are many others. The virus model database that kind of emerged last year. We have 3 already. Then we have a break, and then so the first part is more like the context, and then I will show you some of the results, because since 3 months we are working in Swiss-Prot to identify a function using structure, and it works pretty well, at least for many kind of viral proteins. So I will show you how AlphaFold 3 shed lights on the reovirus. It's an example of the work we're doing. The tools for capsid assembly, so we can, there are emerging tools, and we can completely make a model of capsid, of whole capsid, icosahedral capsids. And more importantly, the structural phylogenetics, because now this is in development and will change a lot, but now it's possible to make phylogeny just based on structure, which is huge because, for example, you can compare capsid proteins for all over the virology. That was not possible before. Okay, so let's start. And we're going to have an introduction to structural virology. We don't have so much structure of viral proteins. It's about 10% of PDB. And some viruses, like here we have SARS-CoV-2, you have about 67 structure of the spike protein. But for other viruses, you have nothing. So for HIV, for influenza, you have a lot of structure for the same protein. But for many other viruses, you don't have so. The viruses are classified, used to be classified based on Baltimore classification. So I put Baltimore was Nobel Prize for reverse transcriptase, not for classification, actually. And this is because at the first we don't have the structure, the few sequence and few knowledge. So we used to classify the virus like that, and they have been classified along this line for like 50 years. Okay. Now, one of the big problems in virology is the sequence diversity is huge. So, for example, diversity in the single family of viruses, it can exceed the diversity of entire domain of cellular life, which is very important. For example, there is a paper here which shows that just megaviridae have much more diversity than the whole archaea and bacteria in the ocean. So, of course, the genome is much smaller, but they are much more diverse, and we have a lot of different proteins with no relation to anything new, or anything known, sorry. So this is a big problem for studying viruses. And here I show you a tool which is called pairwise sequence comparison. It was created by people from the taxonomy classification group. It's in NCBI, and you see, for example, that the sequence conservation among the adenoviruses, so you see within the same species, you have about 90%, but in the same genus, you go down to 60, 55%. And in the family, it's down to 20%. So between families, we have no way you can really make alignment of proteins because the diversity is huge. But you will see that for the structure, it's different. So we have a different kind of structure. Of course, one of the most known and studies is the capsid structure, because viruses are parasites and they are moving between cells. They are infecting new cells. And when they kind of go out of the cell, they protect their genome with a capsid, which is a shell of proteins that protects from environment and nucleases. And there are a lot of different capsids. I show you an insight. So you have a helical capsid, mostly on what we call icosahedral capsid. It's like a 20-faced dice. And it's really interesting because whatever the size of the number of protein, it's all an icosahedron at the end of the day. Okay. So when we look at the viruses, we see that it was published in 2012 that the structure actually unifies the virus universe. So this is the old taxonomy landscape. And you see, for example, in red, so each bar is kind of a type of virus which is different from the others. But in red, you see that the BTV fold of the capsid is shared by many of those. Or like the single gene role of picorna-like is shared by many of those. So you have across all these different family of viruses, which don't have much sequence unicity, you have some folds that are actually concerned. So this already in 2012, we knew that the structure can unify the whole virology, whereas the sequence by itself is difficult. Actually, the only thing we can do is compare polymerases, because polymerases have they have the catalytic site, which is somehow conserved, and we are able across the RNA viruses, for example, to compare just by part of the polymerase, but it's kind of very limited. So what are the virus-specific folds? We have a different kind of folds. We have replication proteins, which could be reverse transcriptase or RNA-dependent RNA polymerase, what are the most, but many other proteins are made for replication. These proteins are kind of very old and usually have no homology in the cellular world, okay? And we have also, I told you, the capsid can be icosahedral helical, so we have a lot of folds. You have like 10-fold of icosahedral capsids and like 20 of helical. And we have also a lot of glycoproteins, like fusion proteins, have several folds, receptor binding, blah, blah, etc. In ViralZone, we have created a page, it is in development, that will record the protein folds. So they are the major one. And since about 7-8 years, folds are used to classify viruses, which what we call the virus hallmark genes, so the genes that are widespread in virology, and fold which are widespread actually. So this is an insight in ViralZone of the, for example, the double jelly roll fold with topology. So the topology is pretty nice to compare different folds, because by using the 3D structure, it's not very easy to catch the eye. But you see, for example, these two folds are really similar. What you put in topology, you see that they are really actually completely homolog one with the other. Okay. So I told you the viral capsids have a cellular ancestor. It was shown by Martin Krupovich and Eugene Koonin, which are really people making huge advance in this field. Very nice studies they're making. So we have, for example, what we call the single jelly roll, which makes, for example, the capsid of picornavirus. There are cellular origins. The double jelly rolls are also cellular origins, so they are not made one from the other, but viruses tend in history to pick up different genes, usually it's enzymes in the cell, and modify them to create their capsid. And here you have the capsid presumably made by herpes and phages, which is a HQ97, which also originated presumably from cells. So when you look at the origin of the viral folds from Kunin and Krupovich in the logic of virus evolution, the theory is that there was a pool of procellular replicants in which you originated the fold like RNA-dependent RNA polymerase, the rep from mononaviria, which is a single-strand DNA viruses, or other kind of replication proteins. And these are unique to viruses. But then these genes have captured cellular genes to make up their capsids. And actually, bacteria have some structure without membrane which look like viral capsid, actually. And these are purely cellular. So it's pretty sure that it happened. And then helicase, which is also a virus hallmark genes, protease for polyproteins, were also captured from the cells. We know that all the protease for viruses were present in Luca pretty much. So therefore are not really something original. But this happened billions of years ago, okay? So evolution went there and the sequence itself is now completely changed and some functions like, of course, now this is a viral capsid and it was not before. So this is kind of what we know on what we have. So you see that folding for viruses is very important because that's what is conserved. In the current classification of viruses, so this is the Baltimore we used to have with double-strand DNA, single-strand DNA viruses, double-strand RNA, and so on, okay? So the RNA viruses are called riboviria now, and they are classified based on a replication fold, so the reverse transcriptase or RNA-dependent RNA polymerase. So that can make trees, I told you, by just using the catalytic site. And so we have a different subset of classification based on this. For DNA viruses, we are using mostly capsids and few other things to classify. So it's completely different way of classification. It's not really good to compare them because here we have just one replication gene. Here we have different genes, like for example, the Duplonaviria, in which we have the tailed phage or the herpes. They're based of HK97 fold capsid, the portal protein assembly, and so on. Here, Varinaviria, in which you have the monkeypox virus, for example, it's based on the double jelly rolls or 2 times single jelly rolls, so fold also that are related. And you have single jelly roll folds there. So you see it's kind of a mix. And sometimes you can have a virus that could be in 2 boxes. So there are a few of them that have been choices. But at the end of the day, we are not classified or something that more universal than just sequence. And this is really making metagenomic studies or anything like that much more efficient in terms of classification. Okay, so the modeling resource I'm going to talk about. So it's AlphaFold 2, of course, we'll see a bit of ASM fold. AlphaFold is based on multiple sequence alignment with predictions. ASM fold is more like a protein language model. And I will tell you about AlphaFold 3, which in my hands completely breaks the code, actually. And AlphaFold 3 is less dependent on the multiple sequence alignment, which is kind of good for virus protein because sometimes you cannot align the sequence itself, but the fold is conserved. So I just show you quickly a MolStar viewer, which is a universal use by people to check. So when you fold the sequence using one of these predictors, you end up with a CIF file, which you can open in MolStar and explore and work with it. So this is basically what you have by default. The structure is colored by chain, so here you have one chain, but you can color it by residue. So you get sequence ID. So it means that the blue will be the N terminus and the red will be the C terminus. So you see actually the topology using colors. And the topology is very important if you want to compare and to figure out their homologies, okay? Because when a protein evolves, it can not change very efficiently the topology. It can happen, but it's pretty rare. So even if the the folding is kind of a little bit different. If you have the same topology between alpha, beta, and so on, it really means there's some kind of homology. So this is a nice way to color, to compare. Many people, when they publish, use this kind of view, which by default is the best. Now, when you model a structure, you will have a local confidence, which is PLDDT. It's a predicted local distance difference test. Okay, so it's scaled from 0 to 100, and it is colored in all the predictors like this. So consider that above 70, it's correct. So you see here, there is a fold that the predictor, it's AlphaFold 2, is much confident about it. But then you have some red stuff, which may be a disordered region. And then this one may be alpha helix, but it's not pretty sure. So it's above 70, so it's pretty good, but not very good. So it allows you to know which part of the model you can trust and which part actually are disordered. That's kind of unusual to find the disorder, because in PDB they don't show the disorder. So many people tend to think that some proteins, like the SPAD of COVID, doesn't have any disorder, when actually there is a lot. Okay, so here it shows that. You also have to know that there is a bias in the training of model, because they're using PDB, what is known. And for example, the spike protein of coronavirus, many of those are crystallized with the receptor, bind to receptor, which means that the disordered that is in the RBD is ordered when bound to the receptor. So the AlphaFold will be trained on something that is ordered, which is not ordered when protein is alone. So there are a few bias like that, but it's not so bad actually, okay? Also, the PLDT is not an absolute quality value, because I told you there can be some bias. You can, depending on the data in which the predictor was trained, there are some. The disorder region will have a low PLDT, but actually you can remove those if you want to study the structure, because they will just make noise. And also, if there are MSA used for AlphaFold, for example, that are underpopulated, you can have a low PLDT, even though the prediction could be good. So it's an indication, it's not an absolute number, okay? So in Molstar, you can also color by confidence. If you go to set coloring, validation, and you can see, so this is a protein of reovirus capsid that I modeled, and you see this part is certainly disorder. So here you can actually select, look at the sequence there, and you can clean up. So what I used to do is, if I want to, for example, make phylogenetic trees, I will remove those. In sequence phylogenetic trees, that depends of multiple alignment, you remove the gaps. Here you will remove the disorder to have a better view of it, okay? So you have also the predicted template modeling score, which is for the whole protein. So if you have, for example, a baculovirus prediction, so you see the local confidence by the colors, which is there. But you have also a PTM for the whole protein, okay? Which could be, gives you an idea. But again, this can be biased, because if you have protein disordered, or sometimes you have different domains, between the domains, the prediction could be bad. And so the PTM could change a lot, but it's a global indication whether the folding software actually is confident and finds similarities of it. So it's pretty nice to have as a global indication. And also you will see that you have the predicted alignment error, PAE, which is extremely important to figure out domains, actually. So it compares the prediction of amino acid placement compared to all others. And within a domain, it's very confident that the amino acids are all equidistant in some way, but between domains, like here, you see that the confidence is very low. In other words, it means for this protein, the modeling program is able to model 2 domains, but it's not able to model how the domains are folded one to each other. So it could be completely different. It could be this part could be turned over. It's possible. So it just gives you this knowledge that of course, the computer gives you one prediction, but there could be several with a joint there. A little look at the tools I'm using. So you have FaltSeek, which is kind of, I can use that a bit like a BLAST for sequence, but it's with the structure. So it's a structural alignment search. It uses structural alphabet and it's extremely powerful. It's just amazing. It just gives you all the domains of the proteins if they are known, like it would be an interpro search. That's really, really fantastic. We will describe the other which are based on phylogenetics later at the end of the course, but you see that they're all using the structural alphabet. So we'll just to see what is it. So structural alphabet, it's developed by Meister Steininger, and it's commented in this publication with Nicolas Matzke here. Actually, it replaces here you have the sequence alignment of a protein. It replaces the sequence amino acid by a code which will describe the feature of each residue and with the neighbor. So there is the software calculates kind of a virtual center of interactions between different regions of the protein, and then you calculate the closest neighbor, and then you change by a letter. So you see it's completely different alphabet than amino acids, which actually describe which kind of neighbor is there. So here, for example, between this beta strand and alpha helix, it will compute some kind of letter that describe that this protein, this amino acid is interacting with this kind of amino acids. And it looks pretty simple when you say it, but it was done with a lot of training. At the end, it is all this vocabulary, which is not so big, and it works really like a charm. Because here you see describe completely the alpha helixes, and you can compare this and be completely independent of the raw sequence, but just dependent on the structure, because even if there are substitution and complementary substitution, the alphabet will take it into account. It will work, okay? So this is used to kind of translate a 3D structure into a linear structure, for which we know how to work a lot to make like BLAST-like, to make phylogenetic trees. So it's completely opening up, it's a kind of revolution, opening up a lot of studies with structure that were extremely difficult before. Because if you compare two structures by looking at all the atoms' position, it takes a lot of computation. It's really heavy to do so. So you cannot make like a BLAST on a very big database. By doing this, it would take years for the computer to compute this. But this, using the alphabet, it's within seconds or 1 minute, you can do it. So it's really changing completely the way we can analyze the structures. So it's called the 3DI alphabet, and it's used in FaltSeek in pretty much everything I show you later. So FaltSeek, I told you, is an extremely powerful tool developed in Martin Steininger lab in Seoul National University. This group is making a lot of new stuff, groundbreaking, incredible, and I'm pretty sure they will continue. So it's really important to know this name if you're going to work with this model structure. So there's a site for FaltSeek, and you can just, of course, you can use FaltSeek by programming if you want. And there is a web also application you can use. You just upload PDB, whatever is a model you made, or it's another PDB, and then you can choose the database. So the database are expanding right now. I used to go to the PDB because PDB is an experimental database, so it's kind of where your knowledge of function is. But there are BFVD, we'll see, which is a virus database, and AlphaFold database, which are devoid of viruses. So you have a way, surprisingly, to compare the kind of 3D BLAST to either cellular cellular level or to virus level here. And you can do it using the 3DI, so the alphabet or TMLine, which is much, much longer. Okay, so you will pretty maybe use that all the time. So fold-seq use the structural alphabet to do so. So we saw that already. So this is what is in the paper. I will go quickly on that. And I show you a fold-seq I made, for example, while looking for capping enzyme of baculoviruses. So LEF4 we knew as a 3-phosphatase, but it was not very clear if the GTPase was there. So I model the LEF4 for which we have no structure. And I use the model. So this is a model I made with AlphaFold 3 actually. I use the model in fold-seq, and you see that fold-seq gives you like 2 domains, okay? And one domain is actually the GTAs, and the other is the TPAs. So you see the mimivirus are the TPAs, which is like this, like a beta barrel, which is found in LEF4, and the GTAs of another virus. So in this, in baculo, they are put together in the same protein, but it's the same fold than other viruses. And you can identify, like you would do for interpro scan, actually. You have an expect value, like you will have from BLAST. But you see, for BLAST, you consider, usually we consider that 10 to the minus 5, which everything that is below 10 to the minus 5 is good. But in my hands, it's kind of different, of course, software than BLAST. You can go much below and find real similarities for viruses, at least for viruses, for the one I looked at. This is done on looking at PDB, so you have at the end of the day a link to the PDB and to the chain in PDB, so you can look what is this chain, because here you see it's PCBV1, so this GTS, but it's not. You have to look in the PDB to figure out which protein it is, especially with capsid, for example, because in PDB you have many assemblies with many proteins, so you have to figure out which one is similar to yours. But it works perfectly. So we're going now to look at the virus model database, because I told you you can use this software to model yourself, your own proteins. But people have made that for pretty much all the viruses. So the first problem in that AlphaFold database, so Google DeepMind and EMBL made an AlphaFold database with all the proteins, but only the cellular proteins, okay? So in the FAQ of AlphaFold, you see that why my protein is not there. Answer is maybe it's the viral proteins that exclude pending improved support for polyproteins. So it seems that they say there are problems with the treatment of polyproteins, but also maybe for ethical reasons, they are reluctant to make viruses, which we can understand. But it's kind of very sad because for virology, it makes so much sense to look at the structure, as I told you in the first part of the course, that we are missing some very big advance in virology here because it's not made in AlphaFold database. So some people have taken the work out, and we have now 3 databases. Our predicted structure, which we can use. So if you want to make a large-scale study of different folds, you can go there. Otherwise, by folding yourself, you need some computer power and sometimes, if you want to make large scale, of course. So we have the first one was made by Norberg and Jennifer Doudna. It's based on RefSec with AlphaFold 2. So you have many proteins. The big fantastic viral database is based on UniRef30, so it's quite different. So there are much more proteins. So these are for eukaryotic viruses. And the most recent one, it was released in December 2024, not yet published, is a Vero3D, and which is based on ICTV sequence of exemplar viruses. And this is human and animal. So the first one I told you is based on RefSec, which is pretty good, and it was it was inserted in ViralZone, so it's available in Model Archive, which is a database for every modeling structure. And you can access in ViralZone, where you have a ViralZone page, for example, for two Ebola virus. You click on 3D models, and you have access by taxonomy of viruses to the models which are there that you can download and use from Model Archive. So this is the first one. It was done with AlphaFold 2. They show in the paper that they can predict function already with AlphaFold 2 of different proteins. So this is, I show you LEF4, but it is LEF3 of baculovirus. And they can show that, for example, there is a structure in pox that can be found in many different other viruses, but are not annotated. We didn't know these genes are having presumably a function related to this one. And you see the different examples like Irido, ASCO, and so on. So they find already with AlphaFold 2 a way to expand the annotation of our proteins by modeling. And then Martin Steiniger made the big fantastic virus database, which is actually huge because they make a UniRef3. So it means that they cluster the viral proteins by similarity of sequence with a similarity of 30%. So it makes a lot of clusters. And many clusters are from singleton, which are maybe a bad quality or maybe just a diversity. It's not really, it's a mix of it. And some non-singleton, here you compare to what is Norberg, the first one I show you. So there are much more, but of course, Norberg database use vertebrate viruses only, or focused on vertebrate viruses, where here, Martin Steiniger, they went for all kind of viruses by near 30. And you can see that by taxonomy, they have a lot of Uroviricota, which is mostly a tailed bacteriophage, which is, of course, overrepresented in the metagenomic resources. So, in my view, it's extremely nice for research and for diversity, but the quality of it is kind of not the best because, for example, they have to cut off the big proteins, restricted the site of proteins, and there are many of these singletons. So, of course, if you want to study phages, that's really perfect because here you have all of them. But if you want to study animal viruses, it would be better. There is a website in which you can check for it for all and browse every structures in BFVD, which is also very nice. So that's a very nice feature because there's all the viruses. Okay, that's really the advantage of BFVD. And now a new one is developed by Joseph Hughes and collaborators in Glasgow, and they call that Viro3D. And what is pretty nice is that they use a virus metadata resource for ICTV, which is ICTV is actually the International Committee of Taxonomy for Viruses. So it has a lot of subgroup. It's a lot of people working since 50 years to figure out the virus diversity and to classify them. And now they are releasing in their database a resource that describe all kinds of virus exemplar for species that's based on the sequence diversity. Also, they add up some virus to represent the diversity. For example, you can have a Lassa virus is extremely diverse. So if you have just one sequence, you will miss a lot of them. So they will give several Lassa virus to kind of respect the diversity of this virus, okay? So the good point is that this is made by a lot of people and experts, so you have a pretty good coverage of quality of it. So they use that as a basis, and they use two kind, USM fold and AlphaFold 2, so they are completely different software, and they choose the best of the two in terms of scoring. And on average, there are much more good sequence from AlphaFold than for SMFold, but not for all the proteins. So they have a mix of it. And at the end of the day, all these models, they have clustered them together. So here you have an insight of the cluster graphical representation in which you see reverse transcriptase are there, or class 3 fusion proteins, and so on. So all this work is also available in a website, which is also made available in ViralZone. So for example, if you go in the virus, I will show you why I show you that. There's a Viro 3D here button. You can check and get access to the prediction of Viro 3D. So it's possible because it's linked to the taxonomy where BFVD is not directly linked to taxonomy. So it was easier to do so. And I show you the provers because it's an example how you can very easily find a new function of protein using modeling. You see, these viruses are negative strand RNA viruses, or a structure like this of the variant. And you see that we have 5 genes, but only 2 we have the function. And the first one we don't know, called U1, U2, U3. Presumably, we know, and this is what makes function hunting in viruses pretty efficient. We know that there must be a nucleocapsid because all the negative strand RNA viruses working with this kind of polymerase, they have a nucleocapsid to protect the RNA, to prevent double strand RNA. So it must be a nucleocapsid in one of these 3 genes. We know that. It's sure. So what does the modeling tell us? If you look, we go on Viro 3D. On Viro 3D, you see model the different genes, except the polymerase, because polymerase is too long, so they are break up according to PFAM. So you don't have the whole part. So that's a big problem we have still with big proteins and polyproteins. But to go back to the point, this is the predictor structure of the U1, okay? And you see, okay, it's an I-fold with, you see, this is low prediction value, high prediction value, and the global score is pretty high, okay? But when I use a full seq on that, yes, when I use full seq on that, what I see is that it's completely similar to nucleocapsid of several viruses in close families, actually. So, and I will show you also what we can do with AlphaFold 3. So from this study on Viro 3D, now it was very easy to me to figure out. You can just download here the PDB on the CIF file. Then you go in FoldSeq, you open it, and you run it, and in 30 seconds, you know, similar to nucleocapsid. It's really fantastic. Easy to do. I just went there because I knew I was missing some data. Okay, I can make that in many viruses in very quickly, efficient way. So that is a kind of a revolution. Then I use AlphaFold 3. And AlphaFold 3 breaks the code for viruses in terms of quality. And one of the reasons is that the way it works is pretty much complicated, but it does not rely on multiple second alignment as much as AlphaFold 2 was. And it's really good for virus protein because I told you we cannot, for example, a capsid or nucleocapsid, we cannot align them by the sequence itself, but just by the structure, okay? So here, the prediction is less dependent on an alignment, which is perfect for the genes we have, okay? And also, of course, it was much more improved compared to AlphaFold 2 in many other aspects. And another thing AlphaFold 3 is able to make is to model different molecules, or you can model multimer also of proteins. So I use that, taking the U1 gene, which is predicted to form a nucleocapsid. As I told you, I've cut off the things that were not really similar to nucleocapsid for the proteins to have something which clear. And what we expect is in this groove, the RNA will be, the genomic RNA will be protected in this groove of nucleocapsid is typical. So by using AlphaFold 3, you can, of course, model your own sequence, which is the trim sequence, but you can model the multimer. And here I have had 8 copies of the protein because I knew it should be found a helical capsid, which of course AlphaFold2 will not find. It will not make, it will make circles, but not helix, because it's not trained to do so. But it just gives you good insight. So what happens if I take 8 of these proteins and I put a RNA inside? So I took the part of the sequence of the virus RNA, which is supposed to be attractive to the nucleocapsid in the leader. Just instead of putting a random RNA, I've put a part of the 3' of the viral RNA, which should make sense. And what makes AlphaFold is actually this. So you see, it makes proteins with RNA, binding them together in the groove inside. So it's also a very strong incentive of we have found the nucleocapsid because what you expect is actually predicted in easy way. And also you can see if you look at the prediction that the RNA, some bases are out, some bases are in, and we know for the work of many nucleocapsids that this is the case. So it may be a way to investigate this by modeling instead of doing, we used to make chemical attack to the RNA to find out which base are exposed or not, or understand the structure. So maybe we are going to a point where we could actually model this and know it just by computation, which is huge. So you see here, we just, by using the data of Vero3D, we figure out that it must be a nucleocapsid, and then using AlphaFold3, we kind of have a good insight of it should be a nucleoprotein binding to RNA and assembling like a helical nucleoprotein should do. Of course, I removed some part of the protein, which was modeled with low confidence. So the nucleocapsid will be a bit more complex than that, but actually the core is there. We should have a break.
Yes.
So now we go in the second part, in which I will show you the work on the reovirus capsids we made to function. We see it's powerful, and this will be published soon. Also capsid assembly tools, and more importantly, structural phylogenetics. We see its development, which is really advancing a lot, but it's so promising for viruses. It's really fantastic. So let's move on. We will start the example with the Colorado tick fever virus, which is a RNA virus, double-strand RNA virus, which is 12 segments. And you see what we knew before this study was just the polymerase. And just the polymerase because there's an active site which is conserved. But all the other genes, we didn't know anything. It was a hypothetical protein. You blast it, you get nothing. So by sequence, they are homologous to nothing in the databases. Okay, which is kind of pity. So Colorado tick fever is a virus from ticks in the North America, which can infect human and induce huge disease like encephalitis in humans. Okay, so we knew by electronic microscopy or many studies that the capsid should be typical of the reovirus with 2 layers of capsids, but we don't know which protein does that. Okay, so I went and folded all of this using AlphaFold3, and that's where AlphaFold3 gained a lot of quality for viral proteins, that it was not as good with AlphaFold2. With AlphaFold2, I was not having something as clear, but with AlphaFold3, you will see that we found many of the function of this protein, at least 4, just by folding this. So here you have the VP1, which is a polymerase. It has a score of 903, okay, the 0.9, which is very good score because it's homologous to the polymerase, don't move a lot actually. The one we knew already, but here we have also a good prediction, and actually it's a capsid of T2, icosahedral capsid, and VP2, nobody knew what it was before, and nobody figured out it would be the capsid. In VP3, you see also have a good score. It's a capping turret with 3 enzymes to assure capping of the RNA that goes out from the shell made by the polymerase, and even VP4 be the outer capsid, and so on. Then we have many proteins for which we have the model structure, which is not so bad. But here, for example, AlphaFold find a good structure, but I don't know what it is. We don't find homologies using fold-seq, not good enough. This one we found is an NTPase, which we knew would be there. So I told you for these viruses, we know that there is a capsid for T2. We know that there's capping turret. We know that there's NTPase, but we didn't know which one. And here we have already, for these 4 genes, we know by modeling who they are. You see some model like that are pretty bad. So there is a bit of beta fold there. And I mean, alpha helix. So often when you get just an alpha helix, you can have a good score because alpha helix looks like any alpha helix. So the computer finds similarity to it, but for function assignment, you don't know at all because there are many alpha helix around and you don't know what it is. Or other proteins, which obviously are different folds. So it can be an insight to what will be interesting to crystallize for study for this virus. So we have the VP2 is completely typical of the capsid of icosahedral T2 proteins, which we expected Colorado tick fever virus to have. So when we make a fold-seq of it, I'm sorry. Okay. So you see the segmented. So this is on the ICTV website in which they have a lot of data about various genes. And they thought it would be methyltransferase, maybe cell receptor, segment 2, VP2. Actually, no. AlphaFold 3 just tells you it looks like a capsid, and you make a fold-seq, and you would see that. So these are complex. I told you for many viruses, you have a complex in PDB with all the proteins. So it's called the RBAP complex, but in this virus, you have the capsid and everything together. So you look at the chain C of the acute complex, and you will see that it is actually the icosahedral capsid protein. And you see here, you have an expected value extremely high. For all the work I've done, it's rare that to have so high. So that's pretty fantastic because look at the sequence alignment. Nobody would have believed that these 2 proteins would be the same, so between the Stipovirus and the Colorado tick fever, but actually the structure folded and predicted by AlphaFold 3 is extremely similar because you see we have an expected value of 10 to the minus 19, so there is no question that there is really homology there. So that's where it's absolutely fantastic and breaking because now these modeling studies allow us to find very distant homologue, so not distant by structure, but by sequence, that we were not able to do before. So it really opens a lot of kind of dark matter in virology that I told you is used to identify phages in some laboratories. But also here, just I was able to identify the function of this protein just by folding using AlphaFold 3. So I show you a comparison of the DUDNA in the DUDNA database, this protein made by AlphaFold 2, in the Viro 3D made by AlphaFold 2 also, and the AlphaFold 3. And this is a Bluetongue virus. This is actually experimental structure. Okay, so you see that in the the first database, the quality is not so high. It's a bit better in Viro 3D made by AlphaFold 3, but you see that the score is still low, where in AlphaFold 3, the score is high, and you have a very good similarity with it. So that's where this gap is huge because because everything is in place correctly, and now you have a really good fold. Compared to here, you have an insight that it may be that some parts are accurately folded, but not others like you see here, compared to that. So okay, this is the same with the side view. So you see, for example, in viral 3D, so from the face of the protein is there. And actually, these capsid protein are like a sheet. You see in experimental, you have they are flat, and on the side they are very thin. Okay. You see in viral 3D, so AlphaFold 2, that the face is pretty much not so bad if you look at it, but if you look at the side, it's completely all over the place. When AlphaFold 3 put that in a thin line like it is in experimental. So here you have really a protein that completely look like, and you can use this model to create a capsid if you want. When this one, it's kind of difficult to believe. It's an insight. You could say, okay, it looks like because you have the topology is pretty similar if you look at the topology, but not as best as AlphaFold 3. Okay. So this is pretty much the same slide. So the same method I use with many proteins. So this is VP3. So this is enzymatic protein, which is attached to the capsid, because in these viruses you have a capsid like that, which is made by 60 dimers of these proteins, and you have at the pentamer side, there are 12 pentamers. You have an opening in which the RNA will be extruded. So the polymerase and genome are inside, and a polymerase will produce RNA that will be extruded. But at the top of this structure are the capping enzymes that will cap the RNA when it goes out from the virion. Okay. So here we model by AlphaFold 2. You see VP3, which was no homology to nose protein and no enzymatic domain. If you look at the sequence in UniProt, there is no PFAM, no interpro, nothing from this protein. When you fold it, you see the two methyltransferases, you see the guanylyl transferase, and they are conserved, and you see the topology is pretty much the same. There is no question it must be the capping protein of Colorado tick fever virus. So by doing this simple work, and because of the high quality of AlphaFold 3 compared to AlphaFold 2, we were able to identify 4 more proteins. We were able to have a structure of VP1, which was not available, so predicted structure. And for the others, we don't know. Okay, so that's pretty normal. In viruses, you have kind of 3 kinds of genes. You have replication genes, which will be the capping and polymerase. You have capsid genes, will be these ones. And you have host interacting genes, which depend if it's a plant host or, and these genes are evolving very fast, the third part. So many of these genes may be involved in that, interacting with the host, with host defense, or allowing for virus budding, and so on. So for this, we didn't find homologies yet. We did that for all the reovirales, actually. And this is an example in a spina rhinoviridae. So you see, I use a most recent publication by ICTV, and I'm working with these 2 experts on this to compare what is known and what is discovered by modeling. So here you have the core proteins, the capsid, outer shell, and here I've modeled a virus for every genera, and even this one's not classified yet. So we see that the polymerase, so in black, it means we have a 3D structure in PDB available with a PDB code. So you see for 3 viruses, we have a PDB structure. We know it out there. But for all the others, we have no ID. In blue, it's where we expected the protein to have the function. So you see the polymerase have blue except in no reovirus. So we expect, but for all the other proteins, it's in red. It means only the modeling has allowed to identify. So now you see that we are adding a lot of functional annotation to all these viruses just by modeling, because here we have homology of structure of this protein, and there is no question for the capping turret that these proteins must have the enzymatic activity we expect there to be. Okay, so it's kind of huge, as you see, and it was just done on one class of viruses, but it could be done on many others. It could be done on phages. So the whole field is open up to expand a lot of our knowledge of function of viral proteins. Also, what will be extremely nice is after some years of this work have been done on the reference, then the new AlphaFold model or new SM-fold, whatever fold you want, may be trained knowing that these folds are presumably have this function, and you could expand further in the dark matter of virology or knowledge based on that. So that's just very good, actually. And in all these new predictions, there was no sequence similarity. Okay. And often you can, for example, for reovirus, I didn't find any similarities. So the virus may be a bit distant from the others or different. Maybe it doesn't have capping turret and so on. We don't know. But you could have expectation if you want to. For example, this is a segment 2 protein of reovirus, which has a very low score. You see, it's orange, really. And so the software is able to predict alpha helix, but how to assemble them together, it's a mystery to it. But if you look at the topology compared to a capsid protein, there is something that look like the last alpha helix a group like that. You have beta sheets at the end and C-terminal, which are also there. And even the bad fold is kind of a bit thin on the side compared to that. So I don't expect this to be the capsid. It may be something different, but if there is a capsid like this, it will be the best candidate. So by doing this work, you can also, with low quality, find some information, some insights of where to look at. Okay, then you need to know experimental or other technique to really figure it out. Okay, so this is about the reovirus. Now I tell you, you can model new capsids that nobody knew before. So can we actually model a completely assembled capsid of the viruses? The answer is yes. So there is a database called Virus Particle Explorer, database 3. It's a third iteration of it, which actually offer many useful views to all the structure of ficosahedral capsids from PDB. And they give a lot of view which is relevant to virology. You see the number of structures they have, it's mostly cryo-EM these days. And you see in the most recent, it's really cryo-EM, a lot. So we have much more. So this is experimental structure, of course, but now we are going into the modeling structure and we will have some of this. I don't know if it's in the database or not, but we will have that in the future. Okay, so this guy are making a lot of works about these structures. So, for example, if you go in in ViralZone fact sheet, for example, for these viruses, cultiviruses. No, I'm sorry, it's maybe OP. Anyway, it's reoviral. You have there a button to click to go directly in Virus Particle Explorer. So you can go because they are classified by taxonomy. So here I go to Arterovirus 1, which is pretty similar to cultivirus. And the idea will be to use that structure as a model to create a cultivirus assembly, okay? Here, this is a description you have in Viral Particle Explorer with a lot of data and tools you can use. You see there are many tools, utilities. I will not go through it. What could be efficient is the new Capsid Explorer, which allows actually, here I click on the Capsid Explorer and you have a MolStar view of the capsid. And here you can choose actually the PDB the PDB chain you want, because you see it's an assembly with a lot of PDB chain, and it can be a problem to isolate them. Here you just have to click to choose one, and you can save, download, and you will have a PDB file to play with. and you can make models like that. So think that I will show you there is an oligomer generator in this website, which is not perfect yet, but already very nice because nobody does that except them. And so they have matrices corresponding to the protein. This is an icosahedral capsid, so T1, T3. So there are different matrices in which they use actually to recreate the assembly. Okay. So you can either make a lot of work with what is in PDB, or you can import your own file. So your own file, you need to have the fold made in the same direction that the one they are. Like it's kind of you cannot take right away what you folded because it will be then not corresponding to the good direction for the matrix. Okay. So what I do is just I pick up one of them, which is in the right direction corresponding to the matrix, and I pick up my full protein and make them match. Then I take over the first one, and I use that to assemble the capsid. So a T2 icosahedral capsid is made actually by dimers, okay, asymmetric dimers, and 60 of them will be assembled into the whole capsid. So I use what I told you. I isolate the dimer from Viro 3D, not Viro 3D, sorry, for Viper 3, and match them to what was extracted for them in the two positions. So the models are now matched in the right position for Viper to make actually the oligomer. Okay, and this is the result I made. So I just put that there. I click and you get at the end, after some waiting, you get a PDB file. You can choose to make just a pentamer or half of the ecoaxial capsid or all of it. So here, so using just the AlphaFold structure we made, we can recreate a complete capsid. So we have to know that the model is simplistic because you just put by geometry, okay, 3D geometry, the proteins together. So if, for example, the protein are not folded completely correctly in angstrom at the border corresponding to the reference, or they are changed in sequence, they may be at the wrong place, or you can have atom overlapping because they just geometry put them together. So it's not perfect yet, but already you can have an insight of how the full capsid would look like, okay? And here I told you the capping turret. Protein is stuck to the capsid, so I've stuck there. And then I made again the program work to create the full capsid, and it is what we get at the end of the day. So you see at each pentamer, we have a turret, which is what happens usually for this virus. So we can model the whole thing. It's not perfect, I told you, because we are not sure, and the border atoms and the size may be a little bit different, but it's already a model you can make, which is great. Yes, okay. So actually here, there's this bubble, and if you look at the sequence, the two domains there are not sure by AlphaFold 2. So this bubble could be a bit up or down. So that's where the confidence I showed you before between the domains are important, because here you are not sure that it's really like a flower. It could be a bit different, because you know for the prediction that AlphaFold doesn't know really this small domain where to put it actually. Okay. So I told you this is kind of simplistic, but there are, of course, people working now very strongly to make it efficient. So this is an example of a publication made by Emmanuel Levy team in Geneva here, in which they are making homo-agglomeration prediction, but by this time computing every atom's predictions and having something very clean and reliable, okay? So they are making multimers already. There is no cathedral capsid yet, but I think if this is possible, it will be possible to make a full cathedral capsid prediction based on model. Okay, so this is, you see, it's but 1 year ago, it's improving. Again, something that will be better at the end. So now the big part, the structural phylogenetics. So as we have no much sequence homologies, but structural homologies, making trees by structure is very appealing for virology. And here you have, again, Martin Steinegger made available a tool which is called FullMason. And it's a multiple protein structure alignment, which is very fast because it's based of the alphabet structure I showed you before for fold-seq. They use the same alphabet, basically, to make a matrix of homologies and create a tree from it. Okay, so you see, this is an interface from FullMason, which is available in the same site as fold-seq. You just have different shows of software there. Here you have fold-seq, FullMason. You just load your all your PDB structures and you run it. So you don't have much yet ways to change the anything in the software. It's really one-click button, but you will see that this is a field with much research by Martin Steinegger and others. So already I could make a tree of, this is a RNA-dependent RNA polymerase for reoviral, the one I figure out. So for this, we already have a tree based on the conserved active site there, but there we can make a tree of the whole protein, structural protein, which is huge. And it pretty much follows what we know about from the site, except for rotavirus. So don't know we are investigating that, but that's really clean. And again, for the capsid, I could make a tree. And so it's the first time anyone in the world make a tree of the capsid of reovirus because no sequence alignment. So it was not possible to do just by structure. So here I have a lot of models. I have few PDBs. I can make a tree and it can pretty much correspond to one we have with polymerase, which we expect. So I also evolve, the capsid evolve together with their own polymerase without much gene exchange, which is what we expect usually. But that's really fantastic and bonkers that you can actually make a tree and compare by using a capsid protein with so long distance. So structure tree allows to see homologies at very long distance. It was not possible before. So before you could maybe, these two proteins, you could have found some homologies, but all of this you could not have access to. Only by structure you can do. But you will see that there is also a problem in that it's extremely powerful for long distance similarity to do so, which is extremely relevant for viruses. But then you are losing a lot of information because here you see this is the alpha helix with amino acids. When you transfer into the alphabet code for 3DI, which look at the actually environment, you are losing a lot of information because here, for example, you can have much more diversity of amino acids than here, okay? And so single mutation, polymorphism, and so on will be lost completely in the study. So it means that this tree is extremely nice, but you are losing some data that will be in this part of the tree by doing so, using this method, okay? And so this is a domain of study. This is a publication in which I really thinking about how to solve this and to make it perfect. So the idea would be to combine a structured tree, base tree, using like FullMason, and to combine a sequence element tree, which will refine, and you could have a much better tree by using the two methods, okay? So this is in development. So there is a program for that called FullTree, which is not yet published, made by in the lab of Cristo Decimo in Lausanne by Dave Moy, and the University of Lausanne. And this is 431. It's available in a SnakeMake workflow. And it really start to use the two of the best, alignment by sequence and alignment by structure and put them together, which is what they have in the bioarchive publication. So actually, in pretty much all the tests they made in many cellular proteins they're working on, full tree gives the best tree. And you see the sequence-only tree and structure tree with different way of making it. And in every aspect, they're using a lot of benchmark of trees, very advanced benchmark, and the combination of two gives the best, actually. So it's something that is still in development, because I know Fault Tree 2 is in development already. Maybe Fault Tree 3 in the next year, I don't know. But so the field is moving a lot. But we know already that it's possible to make trees with both sequence and structure, and to have much deeper trees with higher quality. So that's really good news. So I will conclude this presentation by telling the main points. I mean, advance in structural modeling open new perspective, which are really relevant for virology. It just started because, for example, AlphaFold 3 is just there available since 3 months to people. It's a bit close right now. You cannot make large-scale prediction using AlphaFold 3. You need to log on, and you can make just 20 every day for a single person. But it will improve. Maybe ASM-Fold 2 will recover. We don't know. I'm pretty sure it just started. It's possible to identify homology even if the primary sequence shows no similarities, which is used for virology, because well, there's not a sequence similarity much in this field. And we have new tools that are completely incredible. Tools that we used to have for sequence like BLAST and phylogenetic trees are available with structure now, with fold-seq, fold-tree, and everything that will be developed later. So that's really kind of a revolution, and I was happy to tell you what I learned about this. Thank you.